Cody plans on attending graduate school for either chemistry or environmental science this fall. So let's give a little more.
All right, thanks a lot. Today we're going to talk about stable isotope dendrochemistry and what all that means. And we're going to examine a unique method of tracing nitrogen contributions to an environment with tree rings. So here's kind of a scatter of the things we're going to go through today. We'll start off with talking about dendrochronology and dendrochemistry and what those terms mean. And then we'll talk about stable isotope chemistry. And we'll tie all those things together and look at some research applications that are going on today. And then we'll talk about the significance of doing this kind of research in the global scheme of things. So, dendrochronology. So as we all know, every year that a tree is alive, it grows a new tree ring. And you can count back these tree rings to tell how old the tree is. Hence the study of dendrochronology was born. Dendro is Greek for the word tree, and chronology is the study of time. So dendrochronology examines events through time that are stored in the ring structures of trees. Because a tree can spend many hundreds or even thousands of years in one spot, it can serve as a long-term bioindicator for that environment. Scientists have discovered that trees record any environmental factor that directly limits the growth of a tree ring structure, that directly limits the growth of a tree ring. So they're constantly storing information from their surroundings. So a scientist can use an increment borer, as we see here, and bore into a tree and pull out a sample core like that. Then they can take this sample core back to the lab and analyze it. Typically they measure the widths of tree rings and line them up with different years. So what they'd look at is like, look at a growth ring with a really wide width, and that's really wide, and you could say that the tree had a really high growth year that year, it grew a lot. Or we look at a really narrow ring and say that something limited the growth of the tree that year, such as like an insect outbreak or a late freeze or low precipitation or something. There's a ton of highly developed ways to analyze these core samples. Sometimes we'll take them back and do a chemical analysis on them, and that's what we're going to talk about. So dendrochemistry, the backbone principle behind dendrochemistry is that a tree will store whatever chemicals and elements it's being exposed to in the new growth ring of the year that the ring was formed. So if you can tell what chemicals or elements are available for the tree to take up, then you know what the tree is being exposed to, but more importantly, you can tell exactly when this exposure occurred. So one example that's often studied is like when pollution might be spilled into an environment, you might be able to tell when it happened. Here's a graph that a lot of dendrochronologists and dendrochemists look at with time on the x-axis and element concentrations from the tree rings on the y-axis. So this specific example was done by archaeologists. We're trying to look for exactly when a volcanic eruption occurred. So we can see what they did was they knew what to look for. They knew they were looking for zinc and copper concentrations, and they were able to figure out that this volcano erupted in 1815. Okay, so now we talked about dendrochronology and dendrochemistry, and now we need to start talking about stable isotope chemistry so we can tie all these ideas together. So here we have two oxygen isotopes, oxygen-16 and oxygen-18. They're distinguished by a different number of neutrons, which gives them a different mass. So when we talk about stable isotopes, typically there's one isotope that's far more abundant than the other. So oxygen-16 is usually the isotope, it's usually the mass we think of oxygen having. So approximately, of all the oxygens on planet Earth, very approximately, there's 99.75% of them are in the form of oxygen-16, and only about 0.2% of them are in the form of oxygen-18. So from this information, we can get a really approximate ratio of how many oxygen 18s there are to oxygen 16s on Earth. But this ratio can differ just a little bit depending on the source of your sample or where you're sampling at. And this ratio can also be used to, because they differ just a little bit, to kind of put a fingerprint on your sample, and so you can identify it and learn more about it. It'll make more sense in a little bit, but here's some examples of other stable isotopes that scientists study, such as hydrogen, carbon, and nitrogen. And we can use this fingerprint method on all these different things. So that ratio we talked about, like 1 oxygen 18 to 487 oxygen 16s, we can take that ratio and put it into an equation that makes it really, really simple and easy for scientists to communicate about. It's called the delta isotope ratio. And because these differences and ratios between two different samples, the differences usually happen after the third decimal place in this ratio. So we input it into this equation, and it allows us to talk in whole numbers. It just makes it really easy to communicate the differences between samples. So here's just an example. If you had a carbon-13 to carbon-12 isotopes, maybe that's what we're talking about, and sample A's ratio, once you divide it by a standard, we'll talk about standards in just a second, but the differences are 0.99 and 0.98 in their ratios, which doesn't seem like a very significant difference, but when we put it into this delta equation, we get our values of -10 and -20 for the two different samples. Now, understanding this becomes infinitely easier when we look at real-world examples that like an atmospheric scientist would talk about today. So if you had a sample of ocean water and you wanted to know the carbon-13 to carbon-12 ratio of it, you run it in your mass spectrometer, and you might get a ratio of 0.011142, just an example. Well, you'd also have to compare that to a standard. You can order these standards. They're internationally all use the same one, and the standard has a known ratio of carbon-13 to carbon-12 in it. So you can input your ratio that you found in your sample and divide it by the standard, and then you come up with this delta ratio of -8, this delta number of -8, which is way, way, way easier to talk about than a ratio of 0.011142. So it's all about simplicity, really. This is just an example of how scientists use stable isotopes. We can, Dr. Charles Keeling, he's the guy who's famous for measuring the concentration of CO2 in the atmosphere. He found that the concentration of CO2 gas in the atmosphere is proportional to the delta carbon-13 to carbon-12 isotope ratio. So by measuring this isotope ratio, we can tell how concentrated the atmosphere is with CO2, which, as we know, is very important today. People are very interested today as we're talking about the increase in greenhouse gases and whatnot. Here's just another example of how isotope ratios are used to study the environment. Scientists looked at sediment cores over 5 million years, and they determined the isotope ratio of oxygen 18 to oxygen 16, and they're able to correlate those isotope ratios to temperature to look back and get a record of global temperature for 5 million years. So that was just the introduction to stable isotope chemistry. Now we can start applying the dendrochemistry we talked about earlier to the stable isotope chemistry. So, as we know, trees take up nitrogen as a nutrient. They need it to grow. They take it up and they store it, usually in the form of nitrate. That's the kind of nitrogen, that's the species of nitrogen that's easier for the tree to use. It can take it up in other forms as well, such as ammonia, but then it transfers it into nitrate once it's in the tree, and then from nitrate into amino acids, which it uses as proteins, which will make up the proteins that get stored in cell walls. So nitrate is highly soluble in water. Whoops, sorry. It's highly soluble in water, and which makes it really mobile throughout the tree. So as you can understand, if it took up some nitrate, if a tree were to take up some nitrate in like the 60s and then translocate it throughout the tree, we wouldn't be able to know when it took up that nitrate. So it becomes really important to get rid of all this mobile nitrogen in a tree ring if we're going to analyze it for its chemicals. When you get your tree core sample, you take it back to the lab and we do an extraction. And so that the only nitrogen that's left in these tree rings is the nitrogen that's in those proteins that's incorporated into the cell walls. From the year, it took up that nitrogen from the year that formed that new tree ring. So we take our core sample, we do the extraction, then cut the sample into like one ring chunks, and then enclose those in 10 capsules, or mill them up really fine, and then you enclose those in 10 capsules. And then those 10 capsules go into what's called an element analyzer. It combusts the samples in an oxygen atmosphere and then reduces them with copper at 600 degrees, so you get N2 gas. So all the nitrogen that was in your tree ring is now in the form of N2 gas. And then that gets pumped through a GC packed column to separate CO and CO2. And then your sample, that N2 gas, gets pumped into an isotope ratio mass spectrometer. Ions flow into an ion source. The sample flows into an ion source. The samples are accelerated, focused into an ion beam. The ion beam is deflected by a magnetic field. Heavier ions follow an arc with greater radius, and Faraday cups are placed right where the isotopes we want to see, and right where the masses are that we want to measure. So we have our N2 gas, right? And if it has two, if your ion has two nitrogens, it'll have a mass of 28. If it has two nitrogens of 14, it'll have 28. And if it has a nitrogen 15 in it, or two nitrogen 15s in it, it'll have these mass to charge ratios. So the mass spectrometer can measure all these different isotopes and come up with a ratio for you, which we'll see why that's important. So on to the research. So automobiles, when they combust fossil fuels, when fossil fuels combust, they also release some NO2 and NO gas. It's part of the combustion. And trees can actually, and plants can actually take up nitrogen in the form of NO2 gas. This was done in previous studies under controlled atmospheres through their stomata. So in this experiment, trees were cored and sampled next to a highway in Switzerland. So there's a preliminary study done that measured that delta nitrogen 15 to 14 isotope ratio just from NO2 gas, just from the emissions of vehicles next to the highway at that exact same site in 1994. From the same site where the study we're gonna talk about is coring the trees. So they measured just the isotope ratio of NO2 gas, and they got 5.7. And that's gonna come in and be really important in just a little bit. So the study we're talking about, they sampled trees from 3 different sites. One site was like 10 meters from the highway, one site was about 100 meters from the highway, and the other site was about 1,000 meters from the highway. These are just rate estimates, by the way. This highway was put in 1965 that we're going to talk about. So, as we can see, it's had an increasing number of vehicles per day and emissions. These are just estimates. But so I said they sampled trees from about 100 meters away and about 1,000 meters away. So that's site 2 and 3 here. And these are just measures of nitrogen concentrations in those tree rings, which they sampled like we talked about. This isn't isotope ratios yet. This is just the concentration of nitrogen in the tree rings. And we can see that they have a very stable, they didn't change much from 1920s all the way throughout. The highway was put in 1965. Not a lot has happened. But then if we look at the trees from Site 1 that were right next to the highway, kind of looks like a train wreck on this graph. After 1965, we can see highly increased levels of nitrogen in the tree rings, right next to the road. But we can't necessarily say where this nitrogen has come from. All we can really say is that the trees have been taking up and storing more nitrogen in their trees since about 1965 from looking at this graph. So those same exact core samples were also analyzed for their nitrogen 15 to nitrogen 14 isotope ratios. This is Site 1, Site 2, and Site 3. Site 2 and Site 3 that were distant from the highway, their isotope ratios kind of hung out around -2, -3, something like that. And as you can see, after 1965, the delta values of those nitrogen isotope ratios in the tree rings jumped around quite a bit at Site 1 next to the highway. So we can take our nitrogen concentrations and plot them along with our isotope ratios and come up with, well, we'll take the inverse of the nitrogen concentrations because it spreads the graph out like this. And we can see from this graph that samples that had a really high nitrogen concentration also had a really high nitrogen 15 to nitrogen 14 isotope ratio. And vice versa, samples down here, this is an inverse scale on the bottom here, samples down here with a really low nitrogen concentration had a really low isotope ratio. So what does this mean? This means that the tree isn't just taking up more nitrogen after 1965, as we see from this graph, but it's taking up more nitrogen that has a very heavy isotope ratio, indicating that the primary source of its nitrogen in these latter years was from a very heavy nitrogen source, or one that has a very high nitrogen 15 to 14 ratio. And this points towards the emissions from the vehicles. As we saw from sites 2 and 3 that were distant from the highway, they had a very low isotope ratio, indicating kind of a background. So we can see why it's important, because the trees were taking up, this points towards the trees taking up nitrogen from the trees, setting up from the vehicles, sorry. But there's another way we can show this. There's another way to look at this. We're going to look at what's called a two-member mixing model. So we can say that the nitrogen that's stored in the tree rings is a combination of the nitrogen from the background soil that's in the environment and the emissions from vehicles. So we can make what's called this two-member mixing model. So we know the nitrogen 15 to 14 isotope ratio that's in the tree ring. And we can come up with this equation just saying that what's in the tree ring comes from a combination of what's in the soil and what's coming from the vehicles divided by the total. And we can rearrange this equation, and we can rearrange it a few more times and come up with this equation for a line down here. And this equation for the line is the same equation for this best fit line for our graph. So for our equation down here, I hope everybody see it, our y-intercept is the isotope ratio from the emissions of vehicles. So we can extrapolate our line up here to our y-intercept and see actually what the isotope ratio was from the vehicles, from our tree rings. And that was about 3.1 was the value. So where this gets really interesting is that we could take the information from the 4 trees at Site 1 and put them on the same graph the 4 trees and plot them from site 1. And we can extrapolate, give them a best fit line and see where that y-intercept is. And that's where we come up with this graph, which is pretty cool because we can see the isotope ratio just from the emissions of vehicles as recorded from the trees. And what's really neat is because we can look at the rings from 1994 and calculate their isotope ratio coming from the vehicles as recorded by the tree rings, and we get a value of 4.3 plus or minus 2. And if you remember, as I talked about earlier, the isotope ratio that was the nitrogen isotope ratio that was calculated just solely from the emissions of vehicles from their exhaust back in 1994, they came up with a value of 5.7 plus or minus 2.8. So these two values lie within range of each other, which is really cool because we can say that we can use the trees to measure what inputs are being put into it. So what did we learn? We learned that the auto emissions were providing the tree with its nitrogen from this. We also learned that it's possible to use tree ring chemical analysis to track changes to an environment. And we also learned that tree ring stable isotope analysis can allow us to understand the history of the emissions at a pollution site. So this was just at one site in Switzerland. So we can imagine if there's 161,000 miles of highway going throughout the United States, we can see that that's a lot of environment that that nitrogen deposition could be occurring from the emissions of vehicles and changing the nitrogen cycles of the environments all throughout the United States or throughout the world. So why is any of this important? It's important because policymakers need to know as much as possible about the extent and sources of nitrogen deposition so they can make regulations about emissions. Increased nitrogen deposition can cause accelerated losses of biological diversity in an ecosystem. It can do this because if there are organisms or plants that thrive in low nitrogen environments, they can be wiped out and pushed out by organisms that thrive off of high nitrogen depositions and really high nitrogen concentrations in the environment. Increased nitrogen deposition can also cause losses of soil nutrients and contribute substantially to soil stream acidification, and that can contribute to long-term declines in coastal marine fisheries through processes of eutrophication. So the rate at which nitrogen is entering the land-based nitrogen cycle has already doubled, and it's continuing to increase. So what I mean by that is the atmosphere is full of N2 gas. It's very abundant in the air, but that kind of nitrogen isn't available for plants to use. But when automobiles combust fossil fuels, they're releasing nitrogen from the atmosphere that was unavailable to use in the form of NO2 gas, and it's making it possible for plants to use that kind of nitrogen. So the rate at which that nitrogen is entering the nitrogen cycle is already doubled. It's continuing to increase, and it's changing ecosystems and plant communities. And detailed knowledge of the magnitude of important sinks, such as how plants and ecosystems are taking up this nitrogen, detailed knowledge of this is really lacking. So it's important to do studies like we just did and showed. So that's all I have for today.